Our presenter today, Roser Matamala, received her Master of Science and PhD in Biological Sciences from the University of Barcelona, Spain, and was a research associate at Duke University. She has been a scientist at Argonne since the year 2000. Roser is an ecologist and biogeochemist who studies the carbon, water, and energy cycles at the ecosystem level and the changes occurring due to human factors such as land use and global climate changes. She is particularly focused on understanding interactions between plant roots, soil, and microbes. She is an investigator for many Department of Energy and National Science Foundation projects. Roser has studied prairie restoration effects at Fermilab, and she is currently studying climate adaptation of bioenergy crops. In addition, Roser is developing new sensors for detecting changes in roots and soil moisture. She has published over 50 articles in scientific journals, book chapters, and scientific reports. Her research is contributing to a better understanding of the importance of carbon in soils under wetlands, forests, grasslands, cropping systems, and even Arctic tundra. With this background, it's not a surprise that Roser enjoys gardening very much too. We are pleased to welcome Roser Matamala, who will be presenting What Are Soil Scientists Investigating at Fermilab?
Thank you, Sue, and thank you everyone for joining me to talk about soils and experiments that we are conducting at Fermi. So I want to start, first of all, talking about how soils form. Soil scientists say that there are 5 soil factors that contribute to soil formation. The first one is the parent material. So it's the material where the soil forms from. That could be, in some cases, the bedrock that's in there, but could also be materials that are brought by other areas, by rivers. So that will be sediments from floods or rocks from the rivers. Or it could be brought by ice, by glaciers, or it could be brought by wind. So whatever parent material the soil formed for, that's the first that happens. On top of the rock, you have the process of weathering. Weathering process starts with the disintegration of this rock material by the forces of climate. So, how much precipitation, the amount, the intensity, and the seasonality all influences the disintegration of this rock that's underlying the soil. And besides precipitation, also temperature. So we all know that in cold areas, water that fills rocks, the force of that formation, when it's cold, the formation of ice usually it's capable of break rocks. So that's another factor that happens in climate. Another one is topography. So the slope and the slope of the area is very important. So for example, in a steep slope, you could have formation of topsoil, but that topsoil is very likely to run down and not stay put into that slope. And for the same token, so lowland areas will receive that material that's coming from upwards. The fourth factor will be organisms. So basically, animals, plants, microorganisms, and humans. All of them influence the formation of soil very much. For example, plants, the roots of plants have a weathering, just like precipitation and temperature does, have a weathering effect in which, the growth of those roots sometimes has the force and they produces products that can also break and mix the soil in the material. One of the things that organisms do likely is this production of horizons. So, for example, soil scientists talk about a horizon. The haze horizon is only the soil where the roots are. It's usually a mix of organic material and minerals, and it's usually the topsoil. And then you have, what we call sea horizon, which is mostly devoid of organic material, mostly minerals, and it's down deeper. And then there is time. So time, here you have a time development from 1 to 4 of a soil. The parent material is subjected to all these other factors acting at the same time at different rates and different speeds and intensities. And so time is for all these factors to interact between each other. So you can have the weathering started at 1 all the way to 4, where is the development of a very nice, deep soil, which will be a good soil for farming. But sometimes happens that on top of that soil, there will be a flooding event, a very heavy flooding event, and that soil gets covered with a new parent material, and then this process starts over. So in certain planets, we don't know how many, but this one is Mars, isn't it? And this is the Mars rover, just walking around on the Mars soil. Basically, the other planets have the same as the Earth, isn't it? The four factors that develop soil. So parent material, topography, climate, and time. What's very different on Earth is that we have this biology. So animals burrow inside the soil, they die, their bodies contribute to the soil. Plants and roots that are growing, they also die and then contribute that material into the soil. And then it's happening, as you see here, which is an example of what happened in the fall when the leaves fall. The leaves fall on the ground, and there is all this series of organisms that break down this material into teeny tiny particles. And when that happens, then you have all these microbial communities that will break it down further and create this marvelous soil that you're seeing here, which is what we need for farming. That material contains all nutrients for the plants to grow. Well, you'll be surprised, but there are 20,000 different kinds of soils in the United States. That's easily to say. Usually soils are named by their color, their texture, their characteristics, chemical and physical, and usually they give names by where they first seen. For example, it could be an area, in the name of an area. In the Midwest, we have mollysols. Mollysols are very common around here, and they reflect the parent material, which is in this area, is the wind-blown loss from glaciations very a long time ago. Also, the freezing temperatures during the winter that we have, the humid climate that we have because it kind of rains a lot in here, and then it also reflects the native prairie vegetation and the animals that live in it. Although there are 20,000 different kinds of soils, soil scientists order them, move them into groups, and we have this 12th order of soil taxonomy in which we can agglomerate different soils with similar characteristics so that it's easier for us to work with soil. Here is, I would like you to understand a little bit more why soil is so precious. And this is a video from the Fermilab and Trust that I came across many years ago, and I have shown in several times to middle schoolers. I think it does really present really well why we need to be careful with soil. So bear with me. while the video plays. So you have to imagine the Earth as an apple, okay? If you slice it into 4 pieces, 3 pieces are the bodies of water. Only 1 piece is land. Then you cut this in half, and 3 of that is land that you cannot farm. This leaves 1/8 of the apple. Then the remaining apple you cut in different pieces. Only 1/32 of that apple represents the soil that we have. Now, the American Farmland and Trust says every year the United States loses over 1 million acres of farmland, and we are losing the land ideally suited to grow food. So this is really a big problem that we need to be very careful. If we take humans out of the equation, soil formation in geological times, we know that soil forms 2 to 4 times faster than it's lost. Basically, there is a rule of thumb that says you need 100 years to form an inch of topsoil. So the next time that you dig in your garden and you go about 10 inches, like here, over here, like the yellow line over here, the soil that you are digging up could be about 1,000 years old. Now, when you add the humans to this equation, conventional use of farmland, the soil can be lost 50 to 100 times faster than it forms. And that's just because the cover of the soil is gone. When we put crops on top of it, we remove all the vegetation that was there before. It also, there is erosion, and soil can be washed away when precipitation occurs. And so sometimes there is this kind of streams happening in some farmlands, and that's how soil erodes. There is also an example to visualize what else can happen. So for example, you may all be aware of the Dust Bowl of the 1930s. So in the 1930s, the government was incentivizing farmers to go west to start farming the Midwest because it was very good soil, and the Great Plains. And those farmers were not yet know very well how to farm areas that, are drylands in some places. And so they basically remove all the soil cover, they remove all the prairie vegetation that was there, and they start farming. And so crop after crop, that soil became poor. Now in the 1930s, a big drought hit the area. It was one of those droughts that lasted 9 years. And during that time, huge dust storms started blowing because we have these strong winds in the Midwest. So the winds start blowing the topsoil that was there. And basically you see in this picture over here, this is a picture in the 1930s, where, in this town, there is these soils, this dust storm that's blowing all the topsoil. And you could find soil from the Midwest all the way to Washington, D.C., all the way to the middle of the Atlantic. Now, in 1939, in 1936, rains started, and the dust storms stopped, and people started working differently the land. Now, that has stopped the problems with the soil? Not really. So this is the next picture is a NASA picture from not too long ago, maybe a year or two ago, where you see this dust cloud over here. So this is dust topsoil again being blown away from the land into the ocean. So this is a recurrent problem that's happening all over the world. So what can we do? All right. I don't think we need to say, need to give up. All the soil is going to be gone in 16 years. So we cannot do anything. That's not true. We can actually start talking about, see the soil for what it is a living ecosystem that sustains plants, animals, and us. Very importantly. and treat it as a very valuable resource that needs to be preserved. But at the same time, we need to use it, isn't it? We need to grow food, and we need to grow increasingly amounts of food because there is more people in the planet. So we really need to understand its strengths and limitations when we use it as an ecosystem service. There is already many people doing that. So for farming, for example, there are many people already implementing, many farmers already implementing soil health practices that ensure sustainability of the soil. Some of those are cover crops, not leave the soil bare during the winter. Do rotations, don't have the same crop twice over the same field. Reduce till or go to no till. That's a very important one. And then do composting using manure. Just keep amending, produce more organic material in the soil. Now we need to also use the soil as an ecosystem service. For example, we want it to be used to sequester carbon to mitigate climate change. There is many people that's looking to discover its genetic potential. How can we use the microbial community, the animals, the genetic potential that's in the soil? It's normally said that in a spoon of soil there are millions and millions and millions of microorganisms. So what are the Fermilab projects? Well, I'm going to talk about three. The first one started in the 1980s, and it continues through 2010, and I would say we're still going on with it. And it's about restoration and soil carbon accrual. The second is about the development of soil sensors, and the third is about using bioenergy and genomics. They are related about soils, and I'll let you know how. So if you know Fermilab, this is how the projects are positioned. If you ever go and see those instruments there, now you'll know what they are for. I'm going to start with the restoration and soil carbon accrual. Basically, you're probably familiar with Dr. Robert Betts, who started the prairie restorations in the 1970s at Fermilab. Basically, he sort of worked to discover how to do this using an empty land that was formerly cropped, and started to add native plant seeds to see what it grew, and then as long as it could carry a fire, start carrying fires, do a lot of weeding, get rid of invasive species, and eventually we got the prairies, the restored prairies that we have at Fermi today. And here is the number, the table of the restored prairies at Fermi, that they were restored at different times since 1975, and the last one in the year 2000. So currently, this is a great success. Currently, there are almost 270 native plants at Fermi and many wildlife species. Maybe you have attended the talk yesterday about birds, or other talks in the ecology talks in the open house. By the 1980s, Dr. Julie Jastrow and Michael Miller, both were still working at Argonne, started looking at soil and the microorganisms in the soil at Fermilab. So we had all these prairies that had been restored, and they were restored in a chronosequence fashion. Julie Jastrow was doing her PhD at the time, and she came up with this idea that how the soil gets accrued, how the carbon gets accrued in the soil. Basically, if you pull out a root of any plants, for example, in any plant, you usually see all these little round blobs of soil that are attached to the roots, isn't it? Some bigger, some smaller, some very, very small. This is a depiction of how this looks like. This is the root, isn't it? And then you have these aggregates. We call them aggregates. These aggregates are just, balls of soil that inside you have this plant and fungal detritus, you know, as I was saying before, this organic material that's from the litter. And then you have particular organic matter, you know, pieces of leaf, pieces of roots. And then you have silt and clays. You have also some minerals in here. And then you see that they aggregate in certain forms. They aggregate in microaggregates, so small ones. And then these microaggregates can be united, you know, 4 or 5 at a time, 3, into what we call macroaggregates. So Julie says the transformations and the residue deposition, the transformations of the organic material, deposition from the organisms and from the roots, leads to this formation of aggregates that you can call it an organomineral association with the minerals in there. And that protects the soil organic matter. So if that's true, in a restoration process like the one at Fermilab, can the soil organic matter that was lost during farming can be recovered? And that's the question we were asking. So basically it's no, this is the farmland, okay? And from the time that land is still, it loses carbon every year that it's still like this. There is a curve until sort of reaches steady state that apparently cannot lose anymore. And then if you apply certain conservation practices, it is possible that you go up or you may not. So in about the 1907, about 50% had been lost in farmlands. This is at Fermi. This is a remnant prairie that's still at Fermi. So we went and we sampled some of the soil, and we came up that there was about 12% of carbon in that amount of soil. When we measure a farmland in the same place next to it at Fermi, it came back with about 4.5% of carbon in the same volume of soil. As you can see, this happens to the plow depth. After that, it's the same for both. But the interesting question is, can we recover this organic matter? Well, because we had the prairie restorations, we were going to ask, there are 2 ways to ask this question. How much carbon matter is produced? You know, this is the picture of how this happens. Atmospheric CO2 that's in the air is taken up by the plants, and it's converted into plant biomass. A portion of this mass is transported to the soil, and it becomes soil organic matter. How much of this organic matter stays in the soil? And what we're looking for is to know what's the rate of the carbon accrual in a restoration process. So to do that, we had to team up with atmospheric scientists. Yeah, you'll say, so what does soil has to do with the atmosphere? Well, a lot, actually. To investigate how much plant mass is produced and released to the soil, we use this new technology that's called eddy flux system. An eddy flux system is these 2 over here, you know, is a tower, and at the top of the tower you put these 2 instruments. One is a sonic anemometer that measures the 3D of the speed of the wind speed and direction. It tells you what direction the wind is going and what's the speed of that wind. And this other one over here is an air, CO2, and water vapor analyzer that measures the concentration of those 2 in that air. The eddy flux uses the eddies. So we don't feel the eddies. We don't feel the eddies in ourselves, but the eddies exist. So eddies are parcels of air that move. Some move faster, really fast, and they're small, and some just move across a very large area and maybe move slower. With this technique, with this eddy flux technique, we want to see the amount, the molecule, the exchange of molecules of CO2 and water that goes into above the canopy of an area. In knowing that, then you know how much of the CO2 is going into the plants and the soil, and how much is released from the plants to the atmosphere. So it's just a method to look at that. So we use 1, 2, 3, 4, 5, 6, 7, 8, 9 different prairies that were restored at different times. Now they more than 40 years old, but at the time that we did it, they were from 38 to 16 years old. We also use a farmland that, of course, was over time zero, meaning it has not been restored. And then we use a remnant that we think it was, we don't know how old it was, but we think it was more than 500 years old. So, and this is us sampling across all the different prairies and getting all the soil samples from all the way to 1 meter depth. Then we install 2 of those Ameriflag sites, one in an egg field and another one in one of the restored prairies. One that had been restored about 20 years, so something in between our oldest and our youngest. And this is what we found. With quite a bit of math added to our instrumentation data that come from our instrumentation, we figured out that the net mass production per year for a field that has corn and soybean, corn and soybean, corn and soybean, and corn and soybean, and these are years from 2006 to 2012. Every one of these dots is a measure of those instruments in there. It's telling me how much carbon concentration was in there. It was more if it's positive. So in this axis above the zero line, it means it was released from the plants to the atmosphere. So it was CO2 that was being decomposed, organic material that was being decomposed, and respiration from the plants. If it's negative, down here, this is production. This is corn and soybeans growing. They uptaking carbon dioxide from the air on top, and they building their mass, their biomass. They putting it in mass tissues. So if you look at these, you see the seasonality, isn't it? Winter, there is no plants, so there is only respiration, CO2 production. The farmer plants, whoa, you could even hear sometimes corn growing as fast as it goes. And then corn senesces, isn't it? Then it's harvest, and then you have this long winter where all these are losses to the atmosphere. The next year is soybeans. The soybeans are not such a big plant, so they don't grow as much, and they have less production. And then they harvest, and this happens over and over and over again. So what you can see from this figure then is that the carbon losses are very high in crop fields because there is nothing on the field. And the corn has one of the highest summer uptake of carbon that I've never seen, even possibly more than some trees. Now, if we overimpose what a restored prairie does, immediately you see a lot of yellow over, isn't it? So prairie plants, because they stay there during the winter, I know they senesce, I know they died, but they stay there during the winter, and that reduces the amount of CO2 that's released to the atmosphere. Also, when they grow maybe longer time. So they start sooner than corn or soybean, and then they die more or less at the same time, but sometimes even later. So what we know from that is that there is a large difference between when the plants are active, which is called phenology, and the carbon uptake period is very important for sustaining mass in those fields. So when you look at it, it's the average number of days that the plants are active is greater at the prairie than maize and then soybean. If we were to add all those points on that figure before, and you add them one after the other, this is what the corn and soybean will do. This is how much accumulated net mass will be all right? And this is our zero line now. So if I count year 1 that I measure, year 2 that I measure, year 3 that I measure, year 4, what happens is that the 7-year average that we have in here is that the crops, and total in average for the 7 years were able to uptake about 59 grams of carbon in a year, all right? It's still, they're taking more than they're releasing, but it's kind of small when you compare to what a prairie does. And the yellow one is the prairie. Every year, a lot of this carbon gets put into the soil. So if we then put together our measurements from these towers with our soil samplings and our measurements at the Fermi restorations, we come up with this figure over here, in which from the time that's zero, so the farmland, to the time that there is a remnant prairie, which is over here, all right, at the very end, it only takes about 13 years to look like a remnant prairie, to look like a prairie already. The roots below, the amount of roots that are in the soil, it takes about 64 years. So the prairies right now at Fermi that 40 and 50 years old, they still don't have the amount of roots that a remnant prairie has. Then the microbial community, how much microbes are there? It will take 164 years to recover to a remnant prairie. And the soil goes really, really slow, and it will take about 400 years to become like a remnant prairie. If we partition then our numbers from the towers into where does it go, we can have these numbers over here. In a farmland, only goes 2 liters, you know, what you harvest and the liter that's left, and that's 100%. So 100% of this is left as liter. In a prairie, it's divided by above ground liter, which is quite a bit in there, and then, but remember, then it gets burned during periods of fire. But we have that about 43 grams of carbon per year, or 14-15% of the total goes into the soil, 7% goes to roots, and 5% goes to 2% goes to microbial activity. So to wrap this up, we found in this project that one important mechanism for organic matter to stay in the soil is afforded by the micro and the microaggregate structures found in soils, that the prairie vegetation produces more net mass and soil decomposes lower during the winter compared to farmland, and that farmland needs to apply soil health practices, like, for example, a cover crop during the winter will really help these soils. The amount of carbon that stays in the soil in prairies is about 15% of the net average produced every year. And one optimistic way of looking at this is Fermi soil is already sequestering carbon, and it could be continue to be doing so for hundreds of years to come. So then the second project that I'm going to talk about is the Soil Sensor Awareness Network. So now that I talked to you about that we really have a good measure of the rate of the soil carbon in this restoration, the second question that comes to mind is, so what about water? Because when you look at the system over here in an ecosystem, as much carbon goes down, there needs to be a release of water through the plant. This is in the process that's called transpiration. So during photosynthesis, there is an exchange of water. It's needed for the production of photosynthesis to capture CO2. So do we have enough water? Well, it turns out there are not that many good sensors for measuring water that will give you an idea of a field, you know, of a restored field that you can put in all these restored prairies and look it up. So we team up with engineers and to construct sensors that could be underground and measure water in the field. So this is the schematics of what we wanted to do. we wanted to make a sensor that was teeny tiny or as small as we could make it, that sends information through the soil to an antenna, and it tells us what is the moisture content of that soil. And this is how it looks like, isn't it? This is how we develop. It's a small, all electronics with a battery and an antenna that sends the signal. And this is the final product. So here we are, boring all these sensors into the soil and quite deep to avoid for the plowing. And then we put all these different yellow dots that you see here. Those are the sensors that we added to the soil. Here comes a second problem, isn't it? Each of these sensors produce 48 measures per day. We have 35 sensors, and it measures 365 days a year. So we end up with hundreds of thousands of data points. So now we have to team up with computer scientists, because we need to use artificial intelligence to simulate this field moisture in order to make sense of all this data that's coming from the field. And this is just the way that we've done it. This is a neural network, a machine learning algorithm that's calibrating our signal from the boxes to a reference sensor that we have there to actually produce these. Eventually, we're going to disseminate these sensors into the prairie soil. Here's what you can see. So Fermi, this soil sensor network has a website. It's called Turo, and it's supported by the University of Chicago. So you can go there and look at what Fermilab field is doing on a daily basis. Basically, that's what you're looking in here. That's what's in Turo. And this is our measurements. So we see changes in soil moisture through the signals that these sensors are sending. And we see differences before and after a rain event. So what have we learned from the soil sensor research? That we really need to be developing new technology for soils because we don't have enough. Actually, soil science is quite behind some of other fields. And we need to do that fast because we need to make sure that we know how we're using soil and to help farmers to make good decisions to be more sustainable. Good news is that we got new funding from the National Science Foundation to develop better and smaller sensors. So right now they're not too big. They're kind of small, they're like 5 by 4 inches. But we want to make them smaller. And this new funding is to compare the farm field with the restored prairie to relate soil properties and moisture availability to production of plants and yield. Well, this is the last project. It's a bioenergy genomics research. So this is about looking at the genetic potential of the plants and the soil. There is so many people asking, should we use our land to grow food or fuel? Are some biofuels better than others? Are biofuels sustainable? Will biofuels be able to take care of all of our energy needs? Well, the answer to that is no, but it can offset some. So if we could find a crop that does not substitute food production, makes the most biomass, takes the least amount of energy, and stores the most carbon in the soil, that will be our winner. And here are the 2 field sites that we have at Fermilab. The first one started in 2008 and is 42 plots with 7 plant treatments and 2 nitrogen fertilizations. The 7 treatments are here, which are 4 with switchgrass, which is a bioenergy crop, and it was our model for these experiments. And then we did this big blue stem, in which was a mixture with switchgrass and big blue stem over here. And then we did this other one, so different treatments with different species, plant species, to actually look whether variations in the species that we grow will make a difference. And then there was a prairie mix with many different species over here. Switchgrass is a prairie species too, so it was part of this study too. You see our plots, they were huge, 20 by 36 meters, and we had a lot of fun actually harvesting these fields. And this is what we found. The bioenergy production versus plant diversity and fertilization effects on yield and effects on soil. So the first one over here is effects on yield. These are the 7 treatments that you see, the big blue stem, the prairie mix versus the 3 different switchgrass cultivars. They were fertilized and unfertilized, so fertilized and unfertilized in here. And this is the yield, so how much mass, plant mass they produce. As you see, this switchgrass over here was the one that produced the greatest, but the errors are very small associated to all. So certainly you need to fertilize a little bit because the yields are usually greater, isn't it, in here? But our big blue stem and our prairie wasn't that far from the rest of the switchgrass. Slightly lower, but still a huge production. When you look at that, they are about 7 megagrams of hectare. So yes, we could be using the switchgrass here. Better to use a mixture of switchgrass genotypes, you know, here. And then we look at the soil. So this is data. Now the 3 treatments have been moved together to do the three groups. You know, one that is only switchgrass, then with that one that has all these mixed species, and one that only has big blue stem in it. And what we saw is that this didn't have any effect on soil carbon, but when you grow big blue stem, you have again what we saw at the prairie. You know, more carbon going into the soil. So that was a big eye-opening for some of the bioenergy growers. The second project that we have with bioenergy is some that comes from the biological side. People looking at microorganisms. DOE has this big idea that if we could look at the genomes and identify traits from plants and soils that can be used to improve bioenergy crops and sustainability across different environments. So from the genomes, scale up all the way to farmland, all the way to ecosystems. How to use the power of the genome, whether in the plants or in the soil, to improve sustainability of bioenergy crops. These are fairly recent projects. We have this first one is a latitudinal gradient that goes across Illinois, Missouri, and Texas. So encompasses the continental scale of the United States. We have large experimental plots, 6 by 6 meters. We have 6 cultivars, Alamo, Liberty, Canlo, of ecotype of cultivars that are more on the lowland type, which have different characteristics that upland types, with more tillers and thicker and things like that. So the uplands that we are cultivating are Blackwell, Carthage, and Kevin Rock. And we have the field already set up, and we are looking so many different parameters, whether for the plants, whether in the soil, and whether a carbon in the soil. Here's an example of just green up, for example. So when do the plants start greening up? At different times in Texas, Missouri, and Illinois. Whether we can use traits, green up traits, so how fast a plant starts in the spring to accelerate, to use it as improved sustainability crop by genetic change in that, or 50% flowering. Is there an advantage of certain genotypes by flowering earlier or later that gives an advantage, more yield, for example? Here is us exploring photosynthetic traits. We use this huge chamber. You have to imagine that we have, in this case, 1, 2, 3 pieces, but we can go up to 5 pieces sometimes. And we are looking at how much light the canopy can take. When does the canopy stops photosynthesis and does not produce more mass? When it gets saturated, that will be this point over here. And we have looked at upland and one upland and one lowland, and we already see that there are differences between those two at the end of the season of how much the canopy is saturated. That's an important trait that we could be looking for in order to increase sustainability of these bioenergy plants. Another one will be to reduce ecosystem respiration. Is there one of these ecotypes that works better than others? Well, in this case, we don't see that many differences in here, so that will not be a good trait for us to pursue in looking to improve sustainability. The very last one of this project is what is called the GWAS, which is a genome-wide association study. That means that we have collected native material of different switchgrass genotypes from all over the country, from all over the United States and Mexico and Canada. And then we have planted all of them in a latitudinal gradient, again from Canada to Mexico, in these common garland locations over here. This is a picture of how it looks like. Single plants of each of these genotypes that we selected from all these collections. And we are measuring a lot of different things for these plants. Not just the green up that I showed before, or the 50% flowering that I showed before, but also senescence. When do they die? When do they put seeds? Which ones are better? Are pathogens, resisting pathogens? Which ones produce thicker roots so that they could put more material in the soil for carbon sequestration? So there is a whole bunch of measurements that we're making in every single of these plants, and they are all planted the same across this latitudinal gradient. Here's an example. Here's just a description of the different, how many do they have? And so there are about 327 in these different sites over here. And Fermi, just over here, we have about 342 different plants that are tetraploids, which is a DNA, quadruple DNA in the plant, and 128 of octoploid switchgrass that we are looking at. So comparison across all these sites of all these factors, parameters that we are measuring will tell us what traits we will want to pursue for the genomic potential. So I think finishing up, what did we learn? We learned that increasing sustainability of bioenergy crops, and it comes by implementing new ideas. For example, mixing up different cultivars of switchgrass to improve yield, or putting some big blue stem that actually is much better in putting carbon into the soil. We also have good news for these. We have new funding from the Department of Energy to look at below-ground traits. Those from variations in root structures in between plants, as well as determine soil organisms that are associated to better plant growth. If we could find microorganisms that are associated to certain cultivars that love those cultivars more than others, for example, that would be a way to make that cultivar grow better in certain areas. So I want to give many thanks to particularly Julie Jastrow, Mike Ricketts, Tim Bachtovin, Scott Hoffman, Jeremy Lederhow, and lots and lots of students at Argonne. I mean, if you don't know these, tell your kids. All the national labs have an undergraduate program where the students can come at the national labs and work with scientists for 10 weeks. And, we have at least 20 or 30 students have come through this program. Also, many thanks to Tom Younger at University of Texas at Austin, and Srinivas Balivada and Swati Guha at UChicago. And here is, I don't want to end without showing you that we also have fun looking at soils. these are some of the students from this program. This is Tim, jumping on a pile of switchgrass, and, Julie and I, more serious, just sampling soil. Thank you. Thank you very much.
Okay, thank you, Rosaire. We have a few questions, and I also want to encourage people to type any questions in the chat box because there still are a few more minutes left. So, Rosaire, I'd like to start with a question that was asked prior to your presentation. And you did address this somewhat, and maybe you can say a little more. So the question is, can you please talk about research into regenerative soil building practices? So the person wanted to know what is effective, and secondly, can we shrink the time frame for replenishing topsoil?
Yeah, so I think I addressed it a little bit in one of the slide that said, what can we do? So right now there is quite a current of scientists, farmers, and just interested people that trying to work on soil health, how to improve soil health so that we have soil for the next hundreds of years, isn't it? So it's not all blown out. And there is these regeneration processes that we can do. For farming, as I pointed out, there is already quite a few that people have been thinking of. Cover crops is a huge one. Don't leave the soil bare. Don't leave your garden bare, okay? Just don't leave any soil bare. Anything that has plants on it, that's when things happening. Composting, rotating, no-till, going to conservation practices of no-till, going to conservation practices of reduced till for areas that cannot go to no-till. Things like that. Composting is very important. Using manure, keeping your soil mix, all this. Anytime that you put organic materials into your soil, you are enhancing, you are accelerating the process completely. You are feeding the microbial community in the soil to work for you. and this is an interesting topic because not that many years ago, people didn't think of that, For example, in your yard, people rakes all their leaves. Well, you actually taking away a lot of the nutrients that are, in those leaves. You're taking those away. Luckily, there is a lot of composting groups, for example, that collects those leaves and they do piles of composting. Like, for example, Fermilab has done that for many, many, many years. They collect the leaves from the area around Fermilab and they keep it and compost it to be used and spread in the fields.
Okay, so along those lines, another question just came in about soil restoration, asking about, can mechanical construction of the soil be used to help restore soil and aid in sequestration of carbon?
So say that again, to use the...
Can restoration of soil and sequestration of carbon be accelerated by mechanical construction of the soil?
Okay, so that's an interesting one in the sense that I don't know. I don't think I ever thought of that. Let me think about it. So, for example, when Robert Betts started with the prairies, there is a certain amount of management that needs to happen. For example, weeds and things like that need to be removed. There is a seed bank in the soil for a lot of these non-native species. So I'm not sure whether there could be a way that mechanically you could improve that sequence of restoring areas, for example. For sure, there is a lot of chemical processes that people use in order to remove pollutants from soil, in order to remove things that shouldn't be there, toxins or toxic metals and things like that. And those are chemical processes that occur in plants that are for restoring soils. So I'm not sure I answered that question, but those are the processes that I can think of now.
Okay, thank you. We've got a couple questions about bioenergy grass experiments. One is, what type of fertilizer was used on the grass?
Oh, good question, and catching me on that. All right, I think it was, I think we put it down as solid, and it wasn't, so there is two ways of fertilizing. One is putting liquid fertilizer in the soil, and the other is putting kind of solid. Usually you have a component of nitrogen and phosphorus and others, so like potassium and things like that. So there are many fertilizers available that will give you what you need for a certain soil. If you need more nitrogen or you need more phosphorus or more potassium or things like that. It could also be amendments for pH, changing pH and things like that. So I think what we put in there was solid fertilizer in, I think it was about 60 pounds per hectare, something like that. It wasn't huge amounts. It was, a farmer in a field could put double that amount, for example. And what you need to count on that is that eventually it rains and that fertilizer, trickles down into the soil and into the roots. So that will be, what I'm saying is that there are many, many ways of fertilizing the soil. And so we did use that rate, and we use it solid urea for that, yeah.
Okay, so it wasn't a composting material or anything like that?
No.
Okay. No. And another question, and I'm not sure if you know this, but the question is, does Fermilab collaborate with the biofuel experiments at Ag Lab in Peoria?
Ag Lab in Peoria. We have many contacts. We could be working with some of the people there directly or indirectly. Actually, that project has at least 20 or 30 partners. So on the top of my head, I don't think we do actually work with someone directly from Peoria, but it could be that we indirectly work with somebody with Peoria because there is so many partners involved in the genetics and bioenergy project. It's a huge partnership with many, many institutions.
Oh, I see that our ecologist, Wally Lavernier, just typed in that Fermi specifically does not.
Okay.
So that answers that. There's time for one or so last question. I know we're over time, and if you do need to leave, go ahead. We want to respect your time, but if there's any one last minute question. Well, I'm not seeing anything, so thank you so much for this presentation, and you have a lot of thank yous in the chat box, and we appreciate the outdoor work that's being done at Fermilab. So thank you everyone for attending.
Thank you.